today is the last day for group presentation for Bionanotechnology. So this is our final group for today. So consists of Kasgina, Azim, Soumayat, and also Ummi. So without further ado, this group will will present about drug delivery, and I will pass to them.
Okay, Bismillahirrahmanirrahim. Assalamualaikum. My name is Erieka. Today, me and my friend Azim, Soumayat, and Ummi would like to present about drug delivery, how nano and bionano and drug deliveries can be connected. Okay, first of all, I would like to briefly explain what is nanotechnology and bionanotechnology. As we all know, nanotechnology is defined as engineering and manufacturing at nanometer scales by atomic precision. The example of nanotechnology is nanofibrillation and electrospinning. While for bionanotechnology, it is a subset of nanotechnology where it is more focused on biological part. It is defined as engineering and manufacturing atom level by biological precedence for guidance, and traditional nanotechnology applied to biomedical and biological needs. The example of bionanotechnology is nanowires from DNA. Next. Okay, so in order to gain a feeling of the relative size of nanometer, this table compares the everyday objects with some biological basic blocks using a nanoruler. As you can see here in the first example, it compares the size of glucose molecule with the thickness of human hair. The size of glucose molecule is 1 nanometer and the thickness of human hair is 0.05 millimeter or 50,000 nanometer. From this comparison, we can say that it is incredible the energy needed for our metabolic activities can come from a molecule that is smaller than a single strand of human hair, which is 50,000 times smaller. Next. Okay, so next I will explain about the nano size in drug delivery. So first of all, what is drug delivery? Drug delivery is a method or process of administering a pharmaceutical compound to achieve a therapeutic effect in humans and animals. Next. Okay, in biomedical applications such as drug delivery, size become an important part to see how well the treatment works. When it comes to biomedical applications, the macro size is not good as nano size. For example, the conventional micro-sized drug delivery techniques used in cancer therapy do not work well because first, it do not target the tumor well, second, it gives toxic effects on healthy tissues, and last, it impairs transport to tumor sites. Next. In the past, different ways of giving the drugs were used, for example, through the nasal and oral. But there is problem with this way. The problem is it may give the high levels of drug in the blood. Because of this problem, targeted drug delivery was made. However, the macron-sized delivery vehicles cannot move passively to the cells and pore cells, including the tumor cells with a pore size as big as 380 to 780 nanometer. Because of this problem, targeted nano-delivery system would be the best solution for use in biological applications. In addition, as a drug delivery system, nanoparticles can entrap drugs into their internal surface and or absorb drug onto their external surface. And lastly, the effectiveness of this nano size in drug delivery can be attributed to many factors. For example, because of physical and biological stability, good tolerance between the components, and lastly, it is easy to produce. So that's all from me. I will pass the time now to Soumayat to explain about the different types of nanocarriers.
you open the other one? The next one. Okay. Next. Okay, so beside the factor that was considered by Sister Erieka, we also need to we also need to think about the how the drug can be delivered to the targeted cell without being digested or disintegrated. So the nanocarrier need to be able to integrate both the soluble and non-soluble drug, integrate them inside and be able to reach the targeted cell without being damaged, so it will be more effective, the treatment. Next. So by that, the nanocarrier need to be able to carry both hydrophilic and hydrophobic drug. It also need to enrich the stability of the drug and do not damage the healthy cell. So it's need to not have any toxic effect and be highly permeable, so it's passed through the pore of the cell, and highly tolerable, so it can withstand the high pH and enzyme digestion. Next. And now that we know all the advantage of the nanocarrier, I will talk about the different type of nanocarrier. So here they are placed from the least convenient to the more convenient. So liposome, which has a aqueous core, is the least convenient, since the drug will be there is high chance of the drug to be hydrolyzed, and also it is unstable and can cause drug leakage. The same for the other type, which is nanoemulsion, and the most convenient is the lipid nano particle, which enclose a solid lipid core, and by having the solid lipid core, it will solve those issues because it's produced from a hot homogenization process. So what is the hot homogenization process? So the hot homogenization process, it's a process which we take the bulk lipid and mix it at high share to the active ingredient at a high temperature, which is higher than the melting point of the lipid. So by doing that, the core of the nanoparticle can remain solid even at body temperature or at normal temperature. So by having that, the particle will not have any exchange with the surrounding environment and will have a less disintegration. So it will solve the problem of the leakage, and it will be more stable, and also help in the release of the drug. The release will be longer compared to the two other. And also for the solid lipid nanoparticle, it is coated by a biocompatible surfactant, which make it more stable. Next. So the problem with the solid and lipid nanoparticle is the core is solid. So because it's solid, it cannot incorporate the soluble drugs. So to make it that we want to incorporate hydrophilic drug, we use a polymer, charged polymer, which will be combined with the drug, which are normally positive, and then this complex will be put inside, is compatible with the solid lipid nanoparticle. So that way we can incorporate the soluble drugs. Also, we can use the polymers to coat the whole liquid solid lipid nanoparticle. By that, it will enhance the bioavailability, and it will have a fast action site, and also it will it will increase the stability in vivo, because in vitro it's easier to control, but in vivo it's difficult. So by coating the solid nanoparticle with the polymer, it will be easier to stabilize it, and also to control the release profile of the drug. That's all for me. I will pass it to Azam.
All right, thank you, Sister Soumayat. So now we move on to the applications of the SLNP, or solid lipid nanoparticles. All right, the first, the one that we're going to investigate today, based on our main article of reference, is our guts, or literally our stomach. So usually our stomach is very hard to treat anything within it. There's a lot of example. If you have ulcers within your stomach, or you have any other problems like lacerations or infection within your stomach, it's very hard to target any form of drug within it. So thus, our main cause of concerns today, based on the article, will be ulcerative colitis, basically stomach ulcers. Because of the physiological makeup of the stomach is very hard, okay? It's very hard to treat anything within it, so thus the SLNPs plays a critical role within it, especially when we consider the stomach's ecosystem. If you have a stomach, like I'm asking you guys a question, obviously everyone has a stomach, but just imagine that you guys are a drug. So how do you want to get to something that is hurt over here? So anyone have any ideas? How do you want to go from here to here, assuming that within your travel, you need to go through range of pH levels, you need to go through other microbes, other bacteria, and sometimes you need to even escape what do you call it, the small thing that always catch to you. So these are the three main problems within your stomach. So how do you get there? Anyone have any ideas? Huh? Colgate? Coating. Okay, basically by protecting yourself, correct? So that's a good idea. So basically you want to protect yourself. Imagine this small thing is you. So you coat yourself, right? So but how you ensure that this coating or whatever armor that you have does not break down, and when it goes past through pH, it goes past through these other microbes, sometimes even you know, being dissolved by the stomach. Anyone have any other ideas? So basically when you have problems, usually we break down the problems, right? So basically how you want to combat pH, how you want to combat this bacteria, how do you combat being digested. So any guys have any more ideas aside from coating yourself? You guys can start thinking, because it's not really a very good, easy example, but it's very simple when you break down the problems. All right, anyone have any other ideas? Very what? Very large dose. Basically, you multiply yourself, correct? So you multiply yourself. So whoever goes down first, you're a good sacrifice, something along those lines. But the one, all right, so to make it possible to pass through all this crazy atmosphere, basically if you're a human, if you consider our stomach like a earth, you want to pass through acidic sea, you want to pass through other enemies, and sometimes you want to be passed through from your own death, right? So these are the three main things on our life. But how does this article combat ranging from all these things, right? They consider what happens at the site. All right, what happens at the site is whenever you have a stomach infection, obviously the infections always include your stomach bile or sometimes your stomach juice. You call it stomach juice. Your stomach juice, sometimes other bacteria that is attracted to the infection because they are perforating outside, because the bacteria is moving outside. So basically there's a very specific bacteria that will always move outside of your stomach. So how they do that is consider this bacteria. So basically, all right, the researchers they code this drug within what Sister Soumayat has already explained. They code this drug, and then they ensure that this coating does not break down at certain pH, and would only be broken down by certain microbes. All right, so certain microbes will break down materials. Only certain materials, like for example, you have let's just say we take a microalgae, they only break down CO2, something along those lines, very specific. Right? So when it's specific, it passes through this pH level, basically it can swim through, and then it cannot be digested by either bacteria and also by the stomach. And when they reach there, this specific bacteria will break down, right, this particle and release the drug to the infection site, thus solving the main problem of going through this acidic sea and etc. So this is how they actually solve it. Now I will pass the one to Sister Ummi.
So I will be talking about the second application of drug delivery. Just now, Azim has talked about the colon disease. So here is the ligand modified cell membrane to deliver the drugs to the glioma. You guys know glioma, yeah? Glioma. It is the tumor that grows in the brain. So the nanocrystal drug is coated with the red blood cell membrane, which has been modified by incorporating the ligands that binds to the disease site. Such modification is important so that the drugs will only display its activity at the desired site. Thus, the better anti-tumor impact will be obtained. As you can see here, the ligands attached to the integrins of the tumor cells and also the neovasculature onto the surface of the red blood cell. Actually, there are two types of the targeted drug delivery, which is the passive targeting and active targeting. So passive targeting, it works by the EPR effect. EPR stands for enhanced permeation and retention, where the drug enter the tumors due to the increased permeability of the endothelial blood microvasculature in tumors. As you can see here, the gaps between the tumor cells is larger, so it allows the drug to enter the tumor area. And compared to the normal cell, it is the gaps is smaller, right? So about the active targeting, it depends on the modification of the surface of the membrane by incorporation of the ligands. So it is the advantages, both advantages EPR effect and the specific targeting of the ligands. So here is the zoom by that. Hope you can understand. So back to the previous slide. So back to this case, it is the active targeting drug delivery because it has ligands. So for the Azim part, it is in my understanding, it is passive drug delivery because it has no ligands. So to conclude, the red blood cell is the biological building block of the human body, which is the Bionano element, and it acts as the carrier to control the delivery of drugs, which is in the form of nanocrystal to the disease sites, which is the glioma. All this happen at the nanoscale, so that is how Bionano, nano, and drug delivery is connected. So that concludes our presentation. question. What is the difference between the passive targeted drug delivery and active?
Okay, so your question was what's the difference between passive drug targeting and active drug targeting, right? So passive drug targeting is basically you eat a pill, it goes to your mouth, your stomach, your intestine, your liver, your bloodstream, and then your brain, and then it triggers response by intercepting the signals. But active, when you eat it or when you take it some other way, it goes into some parts of your body, and then it targets the sick part of your body, according to the carrier lah, according to the programming in the carrier. Yeah.
It is a good try. Actually, I asked what is the difference between the passive and active targeting. So the answer is the passive depends on the EPR effect, and for the active targeting, it depends on both EPR effect and the specific ligands. That's it.
Okay, so now we are come to our second presentation. This presentation is by Aiman, Azam, Hakim, and also Firdaus. So they will talk about Bionanotechnology in drug delivery. So without further ado, I will ask them to take the floor.
Assalamualaikum warahmatullahi wabarakatuh for everyone. Have you ever wondered what is drug delivery? Okay.
What is that? Is that like you become a drug dealer? No.
Okay. So drug delivery drug delivery system is the method of how we administering pharmaceutical compound to achieve a therapeutic effect or healing effect for human or animal. Okay, the most common method of drug delivery include the oral delivery, nasal, skin delivery, nasal delivery, and inhalation delivery. Okay, so drug delivery can be divided into two types. The first type is conventional drug delivery system, or we call CDDS, and the second type is novel drug delivery system. Okay, what is conventional drug delivery system? Basically, CDDS is the classic way for delivery the drug. Okay, for the example, oral delivery, we use the tablet or capsule. We swallow the tablet through our mouth, and it will travel through the body. Okay, for the second type, the second example is cheeks or sublingual delivery. We put the tablet under the tongue, and it will dissolve in the blood through the tissue at our tongue. It's same for the buccal. The tablet will be put between the cheeks, and it will dissolve and absorb in the blood. Okay, for the third example is rectum delivery. Suppositories or the drug will be placed inside the rectum of the people, and it will melt at the body temperature to give a quick effect. This method is quite inconvenient and embarrassing, but it has a quick effect. Okay, for the last example is intravenous delivery. Okay, this is like the vaccine. We put the drug, we inject directly in the vein with a sterile injector. Okay, so for the second type is novel drug delivery system. NDDS is a combination of advanced technique, for example, nanotechnology, to form a new dosage form which are far better than conventional dosage form. Okay. For novel, the drug is delivered in such a way that is only active in the targeted area of the body, for example, cancer cell, and it will release in a controlled manner over a period of time. Okay, so for conventional drug delivery, we use tablet, capsule, or syrup. For the novel drug delivery, there are a few type of carrier that will carry the drug. For example, is liposome, polymer nanoparticle, nanosphere, neosome, lipid nanoparticle, and micelles. Okay, for the researcher invent the novel drug because the conventional drug delivery have many challenges. So Firdaus will explain what is the challenge for conventional novel drug.
Okay, so Assalamualaikum. We know that the conventional drug system has been in the industry for so many years, and many people prefer that. But there are certainly some challenges in this system. So some of the challenges are we need to regularly consume this drug in order to make it effective. For example, we need to take two tablets 3 times a day for it to be effective, like paracetamol. In doing this, we are increasing the toxicity level in our body. For example, when we eat 1 tablet, the toxicity level will rise up past the recommended level, and then it will immediately go down. And then we need to take another one, and then we repeat it over and over until the disease is cured. The other side effect, the other challenge is the side effects, which these drugs, certain drugs can cause irritation to our body. When we eat it, when we consume the drugs orally, it may cause irritation in the lining of our stomach, and also vice versa, the acidic environment of the stomach can degrade the drug. So it will decrease the dose and so on. And then the next one, degradation of drug. And then the next one, when the degradation of drug occurs, the dose will decrease. So we need to take much more drug to increase the dosage, and this might cause overdosage, which can cause major complications such as death. Next. To overcome these challenges, Bionanotechnology is used in the novel drug delivery system. In this novel system, we use nanoparticles to carry or deliver the drug into our body. The nanoparticles acts as a carrier of drug into to the intended target, which these nanoparticles can bind to specific sites or specific target, specific cells in the body. When the drug is arrived at the intended targets, only then will it the drug will be released to the effector site. Next. In our selected article, it studies the preparation and characterization of hollow microspheres, nanoparticles for drug delivery in anti-cancer therapy. Next. So there are three components in this nanoparticles in drug delivery system, which is the drug itself, and then the nanocarriers, and then the surface coating outside. So in this paper, the surface coating used is the polyethanol glycol polylactic acid. So one of the advantages of this support carrier is it is a biodegradable amphiphilic natural polymer. Amphiphilic means it is both hydrophilic and hydrophobic. And then it is also non-toxic, and it is miscible with many solvents. So this is the structure of polyethanol glycol polylactic acid. So the properties of hollow microspheres, it has a hollow structure, which it can contain the drugs. It also has magnetic properties, so that it can be controlled externally by the doctors to carry it to the intended sites. And then the hollow microspheres can encapsulate large amount of drug particles. So this is the synthesis procedure of the hollow microspheres, where monodisperse polystyrene is added with iron oxide, and then added with tetraethyl orthosilicate, I think. And then after calcination, we can get the nanoparticles itself. Next. So the properties of the, not properties, this is the results that they get from the paper. We can see here at the A image is the monodisperse polystyrene, and then the B one is the hollow microspheres itself, and the bottom one is the hollow microspheres with the drug inside, and also the surface coating with polyethanol glycol polylactic acid. Next. Okay, I will pass to.
So allow me to expand more on nanoparticles. A nanoparticle as a carrier has sizes between 2 to 20 nanometer. A pill, however, is less than 1 cm. A drug molecule is even smaller than nanoparticle. So scientists can fit many drug molecules in the nanoparticle. And when they feed it into the nanoparticle carrier, they program the nanoparticle to target specific parts of the body which are sick. This sounds similar to GPS. So the nanoparticle, they has these molecular keys for binding to specific sites in those parts. So one of the ways to make Bionano drug delivery work is by encasing them in liposomes. So you can see here, it's similar to in the cell, phospholipid bilayer, hydrophobic head, hydrophilic tail, and it also can contain sphingomyelin which can be extracted from membranous myelin sheath in animal cell, and phosphatidylcholine from egg yolk and soybean, and also glycerophospholipid. And we can also add cholesterol to increase the stability and prevent the leakage of the liposome. Okay, so before, without nanoparticle, when we eat the pill, it degrades when it passes through our body to the brain. So in our stomach, we have a very strong acid, hydrochloric acid, and it can degrade the pill easily, and only left some of it to the next stage. So in nanoparticle carrier, it is safe in the case and won't be degraded at all. After that, it goes to the intestines, and the nanoparticle liposome is very small and lipid soluble, so it increases the intestinal permeability, which means it increases the ability to pass through the intestine wall, and that's how they can pass through the intestine wall very easily. And in the liver, before, the pill has no protection from the enzyme to degrade them, but now the nanoparticle carrier protects them without any degradation. And then it goes to the bloodstream, where all the proteins that can bind to the drug molecule, because drug molecule have many compounds in it, like protein. So proteins can bind with protein, and it can create another compound and degrade some of the pill. But now in the carrier again, it is not degraded, and it reaches the brain. When it reaches the brain, it can interfere with neurons for sending, receiving, and processing signals. When you are sick, for example, when you're sick, you take paracetamol. For example, this one is big, right? Paracetamol. When you eat it, it goes there, and it interferes with the pain, with the signal. When you feel pain, you feel headache or anything, it interferes with the receptor pain, so that you won't feel the pain anymore. And then the nanoparticle is much smaller than this. It's just to show the size differences, right? So nanoparticle is even smaller, but it's not degraded at all. So you can eat it in smaller doses. So in a pill, right, 1 cm is much bigger, billions of times bigger than a nanoparticle. But in nanoparticle, you can eat it smaller doses. And then next. And then the biggest advantage is smaller doses. They also not to need to consume the drug, because this one is when you eat the pill, you eat once and it goes to the peak, and then after some times it goes down, and you need to take it again, and down and again. So when you eat nanoparticle, drug in nanoparticle carrier, it can maintain in our body for a long period of time. So it stays here, like this one, it stays here. The efficacy, the amount that makes it work. So yeah, next. So this can reduce the side effects and increase the bioavailability of the drug. Okay, go on.
We must talk about Hakim. I need the mic.
Sorry.
Now we talk about the future prospect of this nano delivery system. As you can see, the direction that this nano delivery system is heading is towards cancer treatment. So we have advanced, and as before, from the conventional, which is using large dose. Using large dose like pill, and for example, into putting it into a nanomaterial, microcarrier. And the reason we do it why is because nanomaterials have high stability, it has high carrier capacity, and ability to include hydrophilic and hydrophobic molecule, and various administration route. You know, the nose, and then the mouth, and then everywhere else. And then next slide. So like I said before, it's used for cancer treatment. For example, it's used because cancer, right? Conventionally, is treated with surgery and also drug, chemotherapy. And then these drugs, and however, it is very toxic to the body. It's very toxic to the body, and it can cause normal damage to the cell because it has it's not able to recognize the targeted site properly. And then there is also a problem of tolerance. When you take antibiotic and then you don't finish it, and then the bacteria become resistant to the antibiotic. Yeah, it also has that problem. So conventional drug delivery, because of that, it's very hard to properly target the cell and avoid resistance. So where does nanotechnology come into play? Like has been said before, we use the nano delivery drug system, and because it avoids the problem of poor selectivity. You know, poor selectivity, it's I also, okay. It means that it's the ability to recognize the tumor cell. Scientists employ bioconjugation. You know, when you have a the particle, and bioconjugation means you have a particle and then it try to bind, and this is, for example, this your particle, and then it try to bind with ni lah benda, or a biomolecule such as a protein or carbohydrate. And also the conjugated particle bind to surface markers on the cancer cell. No, sorry. Okay. The advantages of this conjugated particle, because it has more volume and also large surface area. Take for example gastric cancer. Sorry. Okay. For example, gastric cancer. They postulated that the gastric cancer come from, the thing called H. pylori. Because, for example, this bacteria, this gastric ulcer, is caused by H. pylori. And then, what the bacteria do, they create microcolonies. Yeah, they create microcolonies. They become bigger, and then when we ignore the ulcer, it expresses virulent proteins such as CAGA and VAC-A that control the host immune system to escape immune detection. So, continuing what I said, and the secretion of versatile bacterial colonization factors and virulent factors. I said this bacteria, and it will secrete those things, the vectors, and then it will lead to the cell damage. So when we ignore the cell damage, and it may cause ulcer and atrophy and probably cancer in the last stages. So they postulated if we detect H. pylori early on, we can prevent gastric cancer. So that's all. Question. I have a question. What is the thing that I just said just now? The virulent factors. Okay. The question was now, what are the virulent factors that were secreted by H. pylori? I just said just now.
The answer is cytotoxin, but I don't know the other answer.
Okay, it's vacuolating cytotoxin. Yeah. Thank you so much.
Now we come to the end of our project presentation. This is our last group, consists of Rohani, Jannah and Nisaa. So they will talk about self-assembly for biosensor making and utilization. Without further ado, I will pass to them.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Today, me and my group mates will present about the self-assembly for biosensor making and utilization. Okay, so this is just a brief introduction to biosensor. So how it works is we would have an analyte, which is something that we want to sense, and then we have a biorecognition element, which is antibody, enzyme, microorganism, DNA, cells, or MIP. So in this case, in this presentation, we will focus mainly on MIP, which is molecularly imprinted polymer. So this will go through a transducer, and then the output system will read the data through the computer. Next. Okay, so this is our main article that we will refer to, which is self-assembly synthesis of MIP for detection of testosterone, the hormone. Okay, next. All right, so I will explain to you what an MIP is. So firstly, as you can see, Okay, so the main theory around this is you would have to polymerize a monomer around something that you want to detect. So in this case, we want to detect testosterone. So the polymer will be polymerized around the testosterone. So it will undergo template polymer is the testosterone. It will undergo polymerization with a monomer. Okay, so this is after the polymerization. So after you get the polymer, you will extract the testosterone, and then there's a cavity with the shape and characteristics which is the same as the hormone. So you can see that it's like a lock and key model. So this is how it will detect the testosterone. So it will go through this cavity to detect it. So and then we will deposit the MIP onto an electrode surface to be tested with a sample and to detect it. So basically, this electrode is commonly used as for sensors like COVID tests, pregnancy tests, and stuff like that. So next, I will pass it to Siti Rohani to explain about self-assembly.
Okay, Assalamualaikum warahmatullahi wabarakatuh. I am Siti Rohani and I will explain about self-assembly in nanostructure. So as we all learned already, self-assembly of nanostructure is a process where atoms, molecules or nanoscale building blocks spontaneously organize into ordered structures or patterns with nanometer features without any human intervention. So the easiest example of this is my cell. So we know my cell have amphiphilic molecule which is have hydrophilic head and hydrophobic tail. So in higher concentration, this amphiphilic molecule will form my cell because the hydrophobic tail is phobic to water. So it will form this structure to avoid themselves from water, and the hydrophilic head, because it loves water, it will be outside of the micelle. And also, the same thing can happen in non-polar solvent, where it is called a reverse micelle. Instead of the hydrophilic head being outside, now the hydrophilic head is inside the micelle, and the hydrophobic tail is outside of the micelle. So this is the animation on how from monomer it will self-assemble to form, as Tanya explained in Monday, from 0D to 1D to 2D and 3D structure. So at first it will form sheets, and then it will form helical ribbons. And then higher concentration and conditions will form nanotubes. So, okay, that's it. You can go to next slide. Thank you. Okay, so, all right. So self-assembly in nanostructures consists of these forces: hydrogen bonding, Coulombic interactions, pi stacking, van der Waals, electrostatic, and hydrophobic interactions. Okay, next. One of simpler example that we can see in our daily life is, do you realize when we eat cereal or anything, they will usually clump together on top of the milk. So what happened is, this is called Cheerios effect. Okay, next. What happened is, because the Cheerio will absorb the water or milk that it is in. So the same concept with what Sir has explained in the class using the frilled paper. Because it is wet, and water attracts water. When it is with another wet Cheerios, it will the water will attract the water inside the Cheerios, so that it will force it to move together, to clump together. Okay, patah balik. Okay, so where's the marker? So what does this relate with our article is because in the article it said the methodology is our polymer, which is polyaniline, will be mixed with polyaniline. Short form is funny. It will mix with the testosterone inside the solution, and then because the articles talk about electrochemical self-assembly, so the rod that will become the sensor will be dipped into this solution, and because there is already testosterone and pani inside, they will self-assemble in this surface. And then this rod can be taken out and washed with ethanol to remove the testosterone, the template testosterone that is on it. So as Jana has explained, it will only leave the testosterone molecular imprinted polymer. So when it is used in tests, when we use it on human urine sample, the testosterone that is existing in the urine will be detected by our molecular imprinted polymer. So that's how it works. Okay, I'll pass to Sister Nisaa.
Okay, so I will continue with the application of both theories. So the article that we review use the conducting polymer sensor-related technology and electrochemically prepared molecularly imprinted polymer. It also uses an electrochemical method to optimize the synthetic self-assembly of polyaniline by coating the electrodes to form testosterone imprinted polymer (TIECP), and the environment that's suitable for this activity is acidic. So when the environment is acidic, it requires an inorganic acid to dope the aniline, to dope the amine groups in aniline. So when the aniline is doped, it will provide a high conductivity, which is good for electrochemical sensing application. And then finally, the random urine samples. And then the spectra of the IECP during self-assembly can be obtained by SEM, scanning electron microscope, and random urine samples were collected, and the testosterone concentration were measured by using the TIECP that we prepared. So next. So basically, there are 4 preparation that needs for this application. First is preparing the reagent, where we need to dope the aniline to get high conductivity, and then synthesis of characterization testosterone imprinted electrochemical conductive polymer, the IECP, and then electrochemical characterization of the IECP coated electrodes, where we need to coat the electrodes, and then the determination of testosterone in human urine samples can be done. Next. So these are the example of urine samples performed by the architect CI 8200 system. This instrument is to provide clinical chemistry and immunoassay testing in a single system. So the concentration of testosterone in the sample can be calculated by using this instrument. So basically that's it from our group. I have one question. So why do we need to dope the aniline?
Yay. We dope the aniline to make it highly conductive for electrochemical sensing.
So that's all from our group. Thank you.